Your career begins here at Satyabama Institute of Science and Technology, a deemed to be university, a nationally recognized and globally acclaimed destination to accomplish your academic aspirations. The Colonel Dr. JPR Research Park, along with the International Research Center, houses advanced research facilities that have 13 centers of excellence, 10 plus technology transfers, 100 plus patents, 10,000 plus reputed publications. The Satyabama Center for Advanced Studies is a state-of-the-art facility built with the aim to cater to the futuristic aspirations of our students with the aid of a metaverse studio, drone labs, makers space, media center, advanced characterization facilities, ICT-enabled classrooms, and supercomputing laboratories. The engineering departments are equipped with sophisticated infrastructure, fulfilling the academic and hands-on requirements of our students. The architecture department is furnished with sophisticated design studios and spacious drawing halls, all of which support the learning process of our postmodern generation architects. The Satyabama General Hospital, along with the School of Dental, Nursing, Pharmacy and Physiotherapy, have advanced clinical facilities. The monumental Central Library houses a diverse collection of books, periodicals and journals. Additionally, it also has an open-air theatre with a seating capacity of 1,000. The Management, Arts, Science, Law and Hotel Management departments comprise of all the necessary and required facilities, creating the perfect ambience for our far-sighted students and catering to their skill set needs with the end goal of equipping them to live their dream. Our chain of laboratories with practical exposure equips our students to build a successful career. Our institution has a team of dynamic and outstanding teachers who are ever reachable to our enthusiastic students, promoting the spaces to learn and places to grow. Greenery around the campus greets and accommodates our students, who are the backbone of a developed nation, edging them on to achieve the summits of their success. Satyabama Institute of Science and Technology.
So good afternoon, everyone. On behalf of Center for Ocean Research and National Facility for Coastal and Marine Research, Satyabama Institute of Science and Technology, I warmly welcome you to another session of Ocean Research Knowledge Series 2026. ORKS 2026 is a continuous expert lecture series that bring together scientists and researchers to share the latest advancements and innovations in ocean and marine sciences. So moving on to the session, today's expert lecture is on jellyfish biodiversity and utilization. And we are happy to have with us Dr. K. Govindaraju, Professor, Research, Center for Ocean Research as speaker. His explain change and marine biotechnology. Over to you, sir. You can now share the screen.
Yes, ma'am. Thank you. Thank you for your
Thank you, sir.
introduction. I'm just
Thank
sharing my PPT.
Yes,
Is
sir.
I think is audible.
Yes, sir. It is audible, sir.
Windows. My screen is visible.
Visible, sir. You can proceed, sir. Thank you.
First of all, I would thank to our Center for Ocean Research and giving me an opportunity to exposure our research related to jellyfish biodiversity and the utilization. First of all, very good afternoon to everyone, and just I'll start my lecture. Okay, you know that jellyfish biodiversity means that. Just a minute, slide is not moving. Yeah, thank you. So already my previous talk, that marine biodiversity is very very uh, uh, wide things when compared to the terrestrial ecosystem. This marine biodiversity, particularly the marine bioresources, a lot of bioresources are there. The previous talks, and many of the scientists are also discussed and explored the many things related to the marine bioresources as well as utilizations. In these things, among these various marine bioresources, today I'm just highlight the jellyfish biodiversity as well as underutilization. That in the marine system, that the ecological diversity already discussed. No, almost 30 lakhs of species are available across in the 71 percentage of its surfaces also. Coming to the molecular discovery, that is a novel in these things, various novel components are novel genes and the various pathways related to metabolic system that's there in this living things. This controlled production in the bio resources, particularly in the controlled production using the fermentation and other techniques like the cell culture techniques that are that biofermentation as well as a culture techniques. Even the aquaculture also, you can see that the controlled production nowadays doing for shrimp productions or fish productions or other crustacean culture, other things are also they are working on it or they are culturing on it related to that is marine bioresources. Coming to the product development for utilization of these and the bioresources, marine bioresources, particularly various that drugs materials or food materials or production of enzymes and various product like that cosmetics or pharmaceutical product. Many of the things are developing using this marine biodiversity applications also. Coming to the social value of this marine diversity, particularly for example the health as well as nowadays rising the blue economy. The economy is very very important. There is a different type of is there, but recent decade that the blue economy rising huge presently is huge for utilization of that marine diversity for various fields. In the our Indian government of India also, they supported a lot of things related to the blue economy for various ministries also. Now the concentrate on the blue economic aspects, particularly in the Ministry of Earth Sciences, Department of Biotechnology, Government of India, and other ministries also, Ministry of Fisheries and Dairy Development. These are all ministers are keenly that involved to develop these our utilization of in the blue economy aspects also. The coming to this landscape of these in the marine bioresources, like the marine animals, the lot of marine animals, that fish, crustaceans, sponges, that other things, other the things like other product like that chitin chitos, that chitin chitos and these polymers also there in these resources. And other part is marine, that is macroalgae, then microalgae, then marine microorganism, particularly is a n- uh, largely available, particularly in the marine microorganism. These things, the bacteria, actinomycetes, fungi, that viruses also they're found in this marine microorganism, means marine ecosystem. These are largely available in the system. Apart from that, the genetic resources like that, the part of genome bar transcriptome base or metagenomics also aspect also there in this landscape. Then symbiotic system, like in the associated things, that marine, that is microbial communities or other, that organism to organism, that is symbiotic system also, that particular, for example, in the coral system or sponges, etc. Many of the things are there in this and the marine bioresources things. Coming to these things, when compared to other ecosystem, that in the marine ecosystem, particularly in the marine bioresources, is a different novel things are available. Why these things are available means is, in this, when compared to other ecosystems, particularly in the marine system or sea system, that is high salinity nature, then high pressures are there. Even though in the 100-meter or 200-meter, 500-meter, 1000-meter, if there are 5000, no, the things in the particular, you just see in the normal, yeah, when swimming pool, you just is a small amount of water is there, you just swimming it. You suppose in this, it's quite large, for example lakes and that things, that the pressure will definitely will increase. You coming to the sea, you think on about the different depth, the different kind of pressures also there, means the ocean current, other things also is there, highly the with different regions also. And as well as the low temperature, that in the surface water is normal temperature also there. In the even that depth is going, the very very low temperatures are, means light sources are there, then very very low temperatures are going. Okay. Even this light, that maybe the 50-meter, 100-meter depth, that some of the lights, slight light sources are there. But in the case of 500,000 means there is no light, only the dark conditions. In this different environmental system, means environmental parameters, the lot of organisms are living. Okay. Depending upon their morphologically, as well as them, even the sick, means the genomically are they adapted in this environment. They meaning a lot of the organisms are living things in these things. So in these things, they are producing a lot of things based on the environmental parameter they're producing, or they protecting itself. There are a lot of chemicals, or a lot of proteins, or a lot of enzymes are secreting for their adaptation, as well as their production of a lot of components, means chemical components also, for their survival of this environment, the extreme day that environments also. That particularly in the marine bioresources, a lot of chemical substances also there, particularly for the halogenated compound. The halogenated compound is rarely seen in the land, means in the terrestrial ecosystem. This type of halogenated compound is largely available in this marine ecosystem only. Then the sulfated, that polysaccharide is available in the terrestrial ecosystem, but in the sulfated polysaccharide is a lot of, for example, in the the ficadine, carrageenan, and the chitin chitosan. These are the polymers, polysaccharides. are only available in this marine ecosystem. It's a largely uh, high value food grade as well as medicinal values are there in this sulfated polysaccharides. You know that you can compare to in this turtle ecosystem that structurally as well as even the organism level is some morphological also different, but in the case of the component structure also is different. You know that is a very complex structure also there in the functional group, means in the functional groups. Even the case of protein, a lot of that complex peptides are there. In the case of chemical compounds, means a lot of functional groups are there. You know that when compared to the other ecosystem, you know that distinct in different kind of that functional molecules are attached in the bioactive components also. Then even though even the lot of organs and the producing the toxin also, when compared to the toxic system, then the marine is a lot of toxic producing organism are there for their survival purpose only. Then other molecules are though in this secretion of enzymes, other things in the extreme environment. Okay, the you know the already largely discussed the utilization of these the marine bioresources, various that food and nutrition or pharmaceutical, and then biomaterials are environmental as well as energy applications, then industrial and productions, cosmetics, etc. Some of only I mentioned here, but a lot of things are they are utilizing for decades and decades. They are using for the various purposes also. You know, coming to the among the various biodiversity means marine ecosystem, you know, the lot of species are available already. We discussed, no, like from the microorganisms to even large animal, even balloons also. In these things, yeah, even in the microorganisms, the different kind of organisms are living in the marine environment. You know, the particular I'm today's the highlighting about that jellyfish. You know that a jellyfish is jellyfish blooms are there. Is the you know that this is the even pollution indicator as well as the climatic indicators. You know that in this summer season the lot of blooms are coming. You know the recently that climate change means a few decades that the climate change is a very big problems nowadays, not only in the terrestrial ecosystem, in the marine ecosystems a lot of changes are there related to the climatic variations. You know that the temperature, that the salinity, pH, everything nowadays has changed in the marine environment. In these things, you know that years and years, a lot of changes are there. Is the weather changes, and a lot of the after 30 years change is the climatic also the change. You know that jellyfish is the in this fish is a whole almost 70% of water only is available in this body. Remaining 30% only that the protein and then other collagenal substances are there. And then other means organic compounds, other things are also available in this organism. You know, the jellyfish, we are not utilizing the food or other things. You know that mainly their peoples are there for the fish capturing and the utilization and other seaweed for cultivation or seaweed utilization, then some crustacean, other things they are utilizing the various purposes, other things. And even know, so we are the marine biologists are mm, ocean related researchers. We know that jellyfish and their utilization, and then what are the things are there. But you know that as other peoples, they don't know the importance of the jellyfish. They know these are toxic things. The peoples are known as the toxic things. Is one type of the swimming, the like the umbrella like structure is there. There are many of the people in the abroad, other things they are using for the ornamental things also, for small jellyfishes are also there. But in the abroad system, there are a lot of toxic jellyfishes are there due to that climate change. Yeah, in even Indian regions, our Bay of Bengal regions also, some of the toxic jellyfishes are blooms are nowadays are rising. So we are every year we are also that monitoring the jellyfish bloom, as well as we are also the studies on the biodiversity as well as utilization. We are also year and year we are monitoring things. So recent years there are a lot of that even in the uh, Pacific regions jellyfish is also nowadays rising here. due to that the climate is change. Even that the Aurelia species that are toxic jellyfishes are found in our coastal regions also there. In this jellyfish bloom, you know that lot of that you already informed know that a lot of global warming, you know, the presently the global warming also very very seriously is going. Then that eutrophication, you know, that changing the environment, and as well as salinity, pH. These are all the changes that the blooms are also raising nowadays. In previous decades, the season wise only the blooming are coming, but recent decades, even the per year, that are the 4 times 5 times also blooming also occur due to that climatic change. You know, in these things there are a lot of work carried out, utilization of these jellyfishes in the, for example, in the venom protein or utilization of various that biomedical applications. We are also working on it, the bioactive peptides for anti-cancer and other applications also. Then you have to utilizing the food and the nutrition aspect. You know that some of the non-toxic, for example, in the chrysaor and other species are also non-toxic. These are also the utilizing for the food, as well as some of the nutrients, the protein rich organism, so we can as well as the collagenous rich things. So these protein as collagenous is very very important for our um, metabolism as well as the aging related things. Even you know that nowadays the lot of marine based collagen for various cosmetic applications also nowadays are rising, no? And as well as the jellyfish we have to utilizing for biofertilizer applications also. Is a lot of is the watering holding capacity is high when compared to other organism. You know, yeah, in the soil applications, a lot of that water holding capacity there, slowly they're releasing the water for the agriculture productions also they are used utilizing. We are also working on it. Even the environmental applications also, recently also we did even plastic biodegradation also there for this jellyfish. This is also we are also explored things, as well as in the microplastic sequestration means microplastic absorption also they are doing for in this particularly in the collagen, as well as in the that protein structures also there. You know that the jellyfish, this already you know that is there's a zooplankton. Is a gelatinous zooplankton. You know that in the things, the classification is the gelatinous zooplankton system. Their contribution of the 92% of the global zooplankton biomasses when compared to others. The 92% of global biomass is there. It's a lot, means a rich biomass is there, particularly the jellyfish. There are a lot of both the positive as well as the negative things also there. We are think on the positive things. You know that a lot of bioactive components as well as lot of that venom, this that peptides are using for various applications, not only the cosmetics, biomedical, even drug delivery, other things are utilizing, as well as various is a indicator, mainly pattern is a climate change, whether the changing in the ocean environment and the blooms are there, maybe the changing in the salinity or changing in the pH or changing the temperature. You know that these changes definitely, that indications are there. The many of the things is a majorly called that jellyfish, one of the major indicator. You know that that these variations occurs in this ocean means definitely that the blooms are there. Then even there is a prominent feeders regularity in the plankton population. You know that the turtles also the eating in the jellyfish. The turtle feed is one of the major feed is jellyfish also. The coming to the negative things means that is in case of the large bloom, no, you know that the fisheries also affecting. The fishery food webs are affecting. The large blooms also there na, the survival of the fishes there is tough, that is survival in this bloom period. You know that you know the jellyfish, that the toxic substances are there. There is a jellyfish venom is the stings are there, no? You know that maybe in the abroad things, a lot of toxic jellyfishes are there. There is marine swimmers are there. That means given the even some of jellyfishes very very highly toxic. Even once they bite, it means via sting automatically, even sudden death also there. A lot of species to species variations also there. And as well as the even the problem in the coastal as well as the tourism as well as the local economics also affecting in the jellyfish blooms. You know that if blooms are there, then the fisheries, when the fish biodiversity also reduce it. Then even though that blooms are there, not automatically the tourism people are not entered. See, these are all the major negative points, other things also. Even these number of the non-indigenous species also nowadays are there due to that in the climatic variations. You know that this is the major classification in this species diversity. You know, so this is the major is the Synadian group. You know, the Synadian group, the lot of three major parts, then the Crypsova and the Hydrosova and the Anthosova and the Cubosova. The Cubosova is the lot of species are there. Even other in the cryptozoals also there, even though a lot of species are available, even based on the structural, morphological as well as genomic variations, a lot of species are available in this our marine ecosystem. Even coming to the our Indian region or Indian coast, particularly in the Bay of Bengal as well as the Arabian Sea, you know, a lot of jellyfishes blooms are there. It means there is a lot of species are coming depending upon the season wise. A lot of jellyfishes are blooming also there. You know that this is one of the study we conducted. You know that that jellyfish biodiversity our NTS coastal region we did on survey also. You know that nowadays everywhere that rising. You know that a lot of some of the species are available in our Bay of Bengal region. But nowadays a lot of new species are also available, are blooming in this our both our Bay of as well as Arabian Sea, even Indian Oceans also. These three oceans are a lot of, and nowadays due to the climate change, the rising blooms are there. Even in the foreign, even available now, so other countries jellyfishes also they coming to blooming in our regions also due to that the changing the climate. You know that in the 1930 to 60, the means the early Chennai region is a Madras region, so it's the Gulf of Mannar region also the service available. But nowadays that 60 to 90, that Ennur, Kovalam and other beach areas are also blooming as also there were. Even 92 to 110 in the other coast like in the Odisha, Goa, that Gujarat also the bloomings also occurred. Now that 2010 to the presently that a lot of almost all the coastal regions, even new species also blooming also occur. As well as our service, even zoological survey of India also every year they are also surveyed, they also explore the species diversity, other things also are going. Then coming to the habitat coverage of these jellyfishes, it means the open host, the open ocean, there are and their blooming answer, even the beach side, depending upon the suppose in the coastal beach side also temperature rising na kuda automatically their blooms are rising. Even in the islands and reefs they know in this environment that lot of jellyfish blue species are also there swimming in this region. Even the ports and harbor, you know that in the even I think over 6 months back, a lot of in the ports that are even our beach side of the lot of blooms are also occur. Even yeah, they see that nowadays even harbor things also that blooming also occur. You know that our Muttam harbor also, even the Kanyakumari, no Muttam harbor also, we see the lot of jellyfish are coming in the things. Even Estuarians, the estuaries as well as the backwater also nowadays. You know that our Muttukad backwater, other things that a lot of jellyfish blooms as well occur. Not only the Kaisor and other jellyfishes are also there. Even these things we are also collected. We'll also that diversity studies. Although what are the species are blooming, we can identify using the morphologically, even molecular techniques tools are we are also using to identify any of the new species also there in these things. You know that the heat map also we did it. You know that the east coast region as well as west coast region. You know this what are the spots. You know that the spots are there. No, these are all the blooms are occurred. Both our east coast as well as west coast also we mapped the one heat map for this jellyfish blooming also. Even that season wise, that monsoon, pre-monsoon, or post-monsoon, even season wise also that the jellyfish bloom has also occurred. That the pre-monsoon, the March and May, is the high temperature period. That even that our ocean surface temperature means SST, sea surface temperature also rising. Then nutrient also reducing the things. Then zooplankton blooms also are there. For example, the jellyfish appears a lot of things in the March to May period. You know that even freshwater things, even freshwater jellyfish is also there. That even some of the river system, that in the Karnataka river system, we are also some suddenly one time that we visited also, even the river jelly also there, small, but small in size, but even the river freshwater system also, that the blooming also there nowadays. Even any other salinity fluctuation, other even ocean current based on the things also, that blooming also there. That in the northeast monsoon, in that October December, automatically slightly that salinity automatically reduced due to that heavy rain, no, that automatically the salinity content also slightly reducing it this time of, that some of the fewer things also coming in the period. But in the case of post monsoon, October, December, that is the apt period for the jellyfish bloom, the particularly our East Coast region. The October to December is the apt period. But nowadays, changing the climatic things, though, and maybe every time the water temperature rising, automatically the blooming also occurred. That you just any other you just monitor the our coastal region. You just ask or discuss with the fishermen. So they told yes, and today's are available. Lot of jellyfish or blooms are there. Likely that due to that the variations of weather as well as the climatic changes are there. The blooming also occur in the different region. Even not only in the Tamil Nadu, Andhra, Karnataka, everywhere the blooms are rising. Then, what are the indicators, the ecological indicators of the jellyfish is very, very important. For example, in the warming sea, already I mentioned, that eutrophication, the low oxygen period. Low oxygen also is the inducer of the blooming also. That predator loss, that overfishing, that also, these are all the ecological indicators of the our jellyfish things. I'm just putting here one image called as a morphological. Morphological of the jellyfish, that is a lot of uh, parts is there, that the bill, then gonads, amps, then other radical canals, ring canals, everywhere is there. That is in the tentacles is very, very important. The tentacle region only, that nematocysts are there. The nematocyst only, that venom is a venom gland. That tentacle is the venom gland, that all the jellyfishes are the venoms are stored in the tentacle region. Means the tentacle region only, the cysts are there. In the cyst only they stored in the venom substances. These are all the just is some morphology of the jellyfish for your just references things. You know that how to identify the jellyfish biodiversity occur in these coastal regions. So that in the previous decades, then that only that after bloom only we identified, different kind of jellyfishes are there based on the morphologically. you know, we identified the jellyfish blooms also. Even molecular things, you just identify using the molecular tool, particularly in the 16s, 18s, that even mitochondrial gene is the common the molecular markers for identification of the jellyfish species. Are even know even the Chrysaora species is a lot, even different kind of Chrysaora species are there, even morphologically same. Even some of the slight changes in the morphology we can't identify suddenly. So color, that color and other structure also, the variations also there, but suddenly we can't see what is the different species of the Chrysaora things. So in these things we have to, even nowadays in the even you can use it for in this the molecular markers. For example, the mitochondrial DNA or synthesization, and we can identify the which species are there are blooming in this particular period. We can easily identify it. Even even nowadays that eDNA technology, ED technology nowadays very good molecular tool, means it's a very good tool to identify the whether these jellyfishes are diversity also occurred in previous years. We can identify using that in this eDNA technology. In this eDNA tool, we can use for the, so what are the jellyfishes are there in the particular region. You just suppose that any Caisora species or any other lineman jelly is available, means there definitely that we can use for this eDNA tool. We can identify the species diversity also. It's a recently emerging for the eDNA tools for identification of the jellyfish diversity in the coastal regions also. There are the already I mentioned of this the molecular tools. We can identify the mm, we also identified a lot of jelly species, particularly the Caisora, as well as in the alignment jelly, that Cyna capillata. These are all the even different kind of Cyna capillata also available, but we can segregate it. We morphologically suddenly you just seen that things is morphologically same, but the species is different. So we utilization of these molecular markers, we identified a lot of jellyfish species, and also we submitted into the gene vacancy based submissions also. And coming to the already mentioned the eDNA for the jellyfish diversity assessment is a recently emerging technology. Even India's very less studies are going. We are one of the group we are working on in the eDNA for the jellyfish diversity studies. Also working, and one of my students also working in our Bay of Bengal region. Was the diversities occurred through in this eDNA technique tools are also. This is some of the um, sort of flow how to that eDNA technology used to identify the jellyfish minas. So coming to the importance of the jellyfish, that the biodiversity, other things we discussed. Coming to that is our jellyfish venom, means that different parts are there, even head part or different part. So one of the major things you already mentioned, no, that it's a nematocyst, particularly in this venom, particularly in the jellyfish venom. So jellyfish is the venom, means it is a problematic in the not only in the human things and other marine organs also problematic, but in this venom a lot of bioactive properties also there. Even species to species are and based on the environment also, that the protein complex also they are changing. That in this jellyfish that venom is a cocktail type, is a different kind of enzymatic proteins, peptides, then biogenic amines, then cytosylizine, hemosynolite. This is a cocktail type of venoms are there in this venom mixture of the jellyfish cyst, nematocyst. Now, that it is a small pulp-like structure. I'll show that cyst also. In this small pulp, this is that the complex molecules are there in this venom complex. That for example in the synodian peptides, means in the acid sensing ion channel toxins, in the cyclolysine, then sodium potassium channel neurotoxin, in the phospholipase. These are even species too, that the combination means in the cocktail also is a differentiate also they even season wise also that composition is also changing based on the climatic variations. That I'll just showcasing the um, nematocyst, particularly I already mentioned, there's this is the nematocysts are there, that is a small dots are there, no, these are all the nematocysts are found in that um, jellyfish. Okay, even just to show that the figure too, this is the like in the pulp like structure. Hmm. In the things you just show that some of the ribbon like structure, like the thread like structure also there. This is the venom for complexes of our the cyst, nematocyst. You just show the next image, just the venom they releasing, like this is the one that like a small thread like structure is coming, this is a venom release in the cyst. These are all the things we identified in this jellyfish venom release. You know that nematocyst is a different kind of nematocyst are there. Species to species are variations also there, even though nematocyst size also variation, shapes also variation, then the combinations also means venom combinations also variations, so depending upon the species as well as depending upon the climate change, they also they changing. You know that yeah, I'm just showcasing the different morphology of the nematocyst. You just show that the means that the anacophore means it is a oval shape or there. Even this is the acrophore means is a oval shape, but shape is slightly different. You know that other nematodes is called like ball like structures also there is the this is the even oval shape, so like it's a different type of shapes are there depending upon the species. Okay, you know that property also is different. You know that releasing property, you know that things, how they are releasing the venom, you know, so that thread like no, the different kind of mechanism also there for releasing that venom also. Even the that structure of this venom, you know that morphologically that complex venom also they're releasing, you know that releasing mechanism also they showed in different shapes are also coming. You just I just mentioning here the some of the species, you know, the Acrometus, then other Pelagia, then Chrysaora species. You know that dominance of the Nematocyst. You told this different kind of already mentioned no the dominant nature of the cyst. For example, in the microbasic Eurotillus, then is the masticophores, plus in the micro basic utilize. Likewise, you just show that based on the species, you can see that number of system shape, then it even functions. You know that Chrysaora species are moderate toxic only. Even the Pelagia and the Nocticulea is highly venomous of things. Then Acrometus is very, very less toxicity than other species also. You know that and based on that is venom complex only, they create a different kind of problem. Our humans as well as other organisms also. You know this neurotoxicity is there, even cardiotoxicity. Likewise, the different kind of toxicity are there, and the depending upon the species to species also variations. And some of the work we did is just to show that this is alignment jelly. You just to show the nematocyst node shape. It's a oval-like shapes also there, that you know that the releasing of the that venom, how they release into the venom. You can see that things. This is a small thread-like structure coming. You know, automatically these are all the venom things. You know that in this last image you just show the that oval shape inside the like the some of the that ribbon like that means on coil like structure also there. No, this is all the venom complex of the our alignment jelly type. This is the first one is for example is the trigger, and the means is also trigger means based on the osmotic pressure, automatically based on the osmotic pressure there trigger that you know that the venom. Then based on the pressure the even that venom releasing, you know, that also that that eversion of the venom also different. You know, some of the venom are coming the tube like structure, or some of the venoms are coming into the voidly as coming, some of the ribbon like structure also coming. Likewise the based on the pressure only they're releasing that venom into the environment. Suppose the sting biting into the human things, you know, based on the species, the effect in the or, human thing or other organism also. You know that even penetration also, even that penetration of venom also depending upon that venom complex. I already mentioned no, that the complex, the different kind of compositions like organic as well as protein enzymes also, these complex also that penetration inside the human body or other organism is very tough. Even some of these is highly neurotoxin, so once the toxic substances are entered very fast into the organism bodies. Even though that when a composition as well as a functional related thing, it just show that things there's a lot of when a composition and the functional things is a lot of different kind of proteins are already some of the research are already purified or explored the toxic venom protein for various applications. Also we are also discovered in the lineman jelly as well as Chrysora, a species we are currently working on for isolation of different kind of venomous substances for various application purposes. You know that here already I mentioned the complexes also. Even that is the pore forming toxins, even the metalloproteases and then phospholipases, they are all the complex of the venom things, even neurotoxin. This is the some other compositions also there. These combination only, based on the combination only, that even highly venom or less venom we can decide it. Coming to the our application or utilization of the results, we have a lot of applications currently we are working. For example, in the anti-inflammatory, even anti-microbial, anti-cancer, antioxidant related things we are currently working. There are some of the research are working on the anti-acaricidal, means acaricidal activity, then immunomodulator also they are working. So almost we are working, our group, our Center for Ocean Research, particularly our group, we are working in a lot of applications also. So we are working for in this jellyfish venom as well as other protein part of jellyfish bodies. I already mentioned that the different kind of Chrysora species also we are collected. We are working in different kind of applications also. You know how the venom or isolation and other important beneficial materials how means extract it means you just collect the jellyfish, then you get isolate the that nematocyst. Particularly in the autolysis process, we can easily isolate the nematocyst. Then you have to extract the
Sir, your voice is not audible, sir.
Sir, please unmute yourself, sir. Participants, there is some technical errors. We will fix it now. Kindly wait.
now it's audible.
Yes, sir. Now it is audible.
In which slide onwards, man? This unmuted, muted.
From
The
this,
box. Okay, okay. Sorry for the inconvenience. Okay.
yes.
So our group also we have collected so different kind of Kaisora species. So we have these are all the some of the morphological structure of these Kaisora species means Kaisora the Sinensis too, so Kaisora and the Kinkuguraceae. So there are the species means of five species, particularly in the Kaisora species we isolated, so it means identified for various applications also. So depending upon the species to species, automatically that venom complex is also changing. Already I mentioned here. So depending upon the that venom complex only that bioactivity, we can work on it. For example, some of the complexes highly antioxidant potential are there. Even some of these things anti-cancer potential are there. Likewise, that based on the complex only. There are in this case of that isolation, the purification of these venom proteins is very very tedious process. So species to species are depending upon the already mentioned that the venom complex based on things only. We can optimize every procedure. For example, in the collection, isolation, extraction, and characterization. These four steps we can optimize the each and every species, not only Skyzora and other species like lineman jelly or other species also. We tried, for example, lineman jelly as well as in the Kaisora species, we can different kind of extraction also we can change. So depending upon that pH, that even buffer system, everything we can optimize for the extraction within the protein. Then only that venom protein, then only we can easily characterize as well as that application as a picture we can decide it. Even the characterization also, this is a common characterization. I already mentioned no that even common that total protein content and other protein separation technique we can using easily we can characterize in the for basic things. For example, in the advanced thing we can using for the protein separation using the protein purification system. In the for example in the acta the related things we can purify it, we can fractionate and purify for further applications also. So, for example, I am just here mentioned here. This is the Kaisora species nematocyst. You just see that the shape of the cyst also variations also there. You know some of the oval, even say same Kaisora is the same species, but in the Kaisora, each species like for Kaisora sinensis or Kaisora fenensis, likewise the species to species that the morphology of the cyst also variations also there. Then only I already told no. So depending upon the morphology or complex only that application aspect also change it, as well as the discharge of the venom, discharges of venom based on the shapes only, as well as I already mentioned the osmotic pressure. These are the important things for the that venom release as well as the toxic is even the moderate toxic and the very less toxic of the venom proteins. You know that we did it on microscopic structure also know that the fluorescence microscope. We just seen that even that surface means that the cyst outer layer shows the emission properties also. There is just the blue color emissions also there is a based on the chromatophore the hypothesis that the blue chromatophore are means like emissions also there in the system membranes. Yeah, I already mentioned this is for example the common technique that is this page you can utilize for this the separation of the proteins. So how many bands are there based on only that how many molecular weight of the proteins are there in the crude venom also. We did it even fractionation also we did it. Even just I just checked it in the crude venom itself. You just show that that is a Bradford protein. You just show that depending upon the species that protein uh, values are also changed. You know that particularly for example some of the things is a 1.3 mg per ml, sometimes is a 1.6 for mg per ml, some of the species 0.6 mg per ml. Likewise that the complex is variations species to species. You just show I'm just showing that the one of the species that is Kaisora species I did the antioxidant system. You just show that variations also there, even that in the species to species that the property of the antioxidants also change it with different antioxidants like DPPH or lipoprotein or nitric oxide, that activity also changed. That based on the venom complex as well as a protein complex and then some other molecules complexes also. Just I also we are worked one of the things means that a few species anti-cancer property also. You can see that even based on the dose dependent activity, even different species, then the activity also so changing. So depending upon the dose dependent as well as the species to species, the activity also changed for the anti-cancer property or antioxidant or other property also. The IC50 values also the changed. You just see in the graph. So and so I'm just mentioning there is time as well as dose dependent killing aspects depending upon the species to species variations also there. Even though that we are also utilization of these venom protein for making the nanoparticle preparation for drug delivery applications also. That drug delivery is one of the important for the proper target to reach the drugs. We need to see some of the vehicle for to deliver the drugs in the particular site. In these things also we did that utilization of these venom proteins we can for the preparation of the even gold nanoparticles for the drug delivery applications. That the complex means that extraction process also we have changed depending upon the species. These are all the conditions we did it for the venom extraction. That reaction of the metal precursors, for example the gold chloride, then incubation period also different, as well as the this is a that I think two different species, to see the two different type of the absorption property of the nanoparticle, then as well as the size of the nanoparticles are also vary. Even the applications means the activity also changed. It's just to show that they did the confirmation of the that gold nanoparticle using the XRD. You can see that even the that structure of the crystal structure also slightly changed depending upon the species also. And further also we can that stability studies also we did it. And also we also which molecular weight of protein are responsible for the um, reduction as well as capping of the nanometer also. We did it. You should show the low molecular proteins means almost 27 kd of proteins is responsible for the that synthesis of the gold nanoparticles also we confirmed it using the STS page. Even biosafety screening also we did it using the um, Ek 293 cells also. You just show the crude venom. Even extraction already I mentioned, no, even the extraction procedure also that toxicity also is very, there's a PB extract is very like toxicity. Even the distillate extract is very high toxicity. Even not only the species to species, that PBS distilled water as well as marine water extraction, these are the things also changed the species to species of the venom extraction. Even some of the species like in the iron alignment jelly is the best for PBS extract. Likewise the Kaisora already mentioned. Next is so depending upon the thing we can optimize those things. Then only that venom extraction also coming into the into the extract. You just show the crude as well as the distilled water, then PBS. That also we did it for the different concentration of the activity. Just to show that depending upon the extraction procedure using that PBS or any other distilled water or marine water also, that activity also that variations also there. Given toxicity also. Likewise, that anti-cancer property also we did it. Given different two type of extraction, known as distilled water as well as PBS extraction. That distilled water also that compared to PBS, the high anti-cancer properties also we did this. These are all the optimization. These are all the fully the optimization of the different kind of jellyfish species. You can which is you just working on atomic first you can optimize. Or you take it out with any other jellyfish species, you can optimize all the parameters even from collection to application aspect. Okay, this is some other species I already mentioned as acrometes, and that is a different kind of species. These also we can tried for the isolation purification and some of the applications aspect also. This is the life morphology of the that Kaisora is sorry, that acrometer species. Just to show this white color, that the tentacles and other body parts also, this is height, it means size as well as length also, that variations also there depending upon the species. You know that in lineman jelly, the length of tentacles or length of uh, other parts also is differentiated also there. You can see that is the oval shape of the nematocysts are there. The releasing, like thread-like structure, no, that releasing of the venom, the based on the pressure. I already mentioned the pressure as the complex of the things. This species also we did it for some of the drug delivery applications for various things. You can see that even the based on the concentration, you can see the morphology of the nanomaterials also changed. You know that a spherical only we can utilize for the drug level application, but other shapes we can utilize for some other application. For example, in the mm, anti-cancer treatment, nowadays using the triangle type of nanomaterials for the anti-cancer treatment for the radiation therapy. That triangle shape of the particles is very good when compared to spherical. Likewise, the research are working on different kind of applications also. Then even we did this normal cells as well as cancer cells. Also, we did it for the Acaromata species of anti-cancer activity. Already I mentioned that even the variations also there depending upon the concentration, they are depending upon the extraction, or depending upon the species to species are there. And last some of the or a few last one application, a lot of things are we are working. But one of the important things that we did on that is polyethylene degradations. Means we did it in this using that jellyfish venom proteins. Means the biodeterioration properties also there. Initially, we studies the different kind of that changes in the plastics. One is called in the biodeterioration, then the biofragmentation is also there, then bioassimilation, the finally that mineralization. These processes are occur for the split of the polyethylene degradations. These are some of the characteristics we did in the FTR, the changes in the polyethylene compositions. This same morphology, we did it for the changes, the normal things, even this different concentration of the mm, venom things, so automatically that biodeterioration property also occurred. We just did it. Some of these normal first contrasting microscope, and this is for the electron microscope. You can clearly see the biodeterioration property of the jellyfish one things. You know that even in this coming recent the marine biotechnology portfolio, you know that these are all the different type of applications. There are a lot of applications also there. I just a few of the things I mentioned, but we have working are different kind of aspect in the case of collagen for wound healing applications, even gelatinous substances for drug delivery application, even microplastic sequestration also there in the mucous protein of the jellyfish. That also one of the students are working. The capturing that not only I am asking microplastic, even the heavy metal also they capturing the mucous protein of the jellyfishes also. These are all the some of the importance of the jellyfish applications. We did on anti-cancer molecule. We isolated in the nematocyst region, as well as the venom proteins also. We did it on novel protein for anti-cancer property, and a few of the working for other applications also, like the immuno modulations or the other things also. We are working on it for different kind of jellyfish species also. You know that future research, you know that many of the research are there. I think there are many of the students also there. You can think on it. It's a different kind of biodiversity also in the environment, the marine environment. Different species are there. This is the means largely they are not utilizing the common people's mean many of the things, because this is the minas of the ocean. Okay, is the like weed like the things that, but you can use this waste to wealth, like the aspect. You can utilize those that means jellyfish. You can utilize for different kind of even food. The already and the acrometer species, you can use you can easily consume it. It's high rich protein as well as high collagen substance. You can utilize those protein automatically is a very non-toxic, not toxic in nature in the body part. You can use in the head part, it means in the umbrella region you can utilize for the even food purposes also. It's a rich of proteins available when compared to other plant-based proteins also. So these are all the thing aspects we are working on it, like in that venom proteomics aspect, as well as the how the mechanisms, actions of different species by species venom proteins are also there for the action of the venom, as well as the AI, that artificial intelligence based also we are working for how the species venom compositions of variations and what kind of application we can apply. Likewise are also we are working. Then even synthetic approach, even you identified another novel protein, means you can working on the synthetic of protein, synthetic protein or synthetic molecules based, you can work on it. These are all the major gaps are also there in this when jellyfishes, not only this particularly venom and other things also. You can utilize the food, you can utilize the you can fertilize it, little things. You can collect the jellyfish things, you can chop it, and you can dry it, and you can utilize for the fertilizer process. A highly water holding capacity, you can the soil moisture they can kept it. So large of the abroad things they are also working. We are also working with I tie up with the agriculture university for this aspect also. Even some other aspects also we are working anyway, due to the time constriction. So I'm closing that talk. Oh, okay. So the peoples are think on it. Okay, there are a lot of things are though you can. So the different even this is the slightly waste material. They are not consumed much when compared to other fish, shrimps, others also. So in these things you can utilize those things for beneficial applications. So I am the just discussing about a few of the things, but a lot of other applications also there. Even you can working on it cosmetic majorly the cosmetic related things and other even environmental applications also we can work on it. So these are all the aspects are also there. So even also we have also this that present and the recent and the academic year, in the present academic year also we started this MSc marine biology course for expanding our research outcome and other things to the students. So we have launched the MSc marine biology program for different kind of aspects, not only in the theoretical other things. So research oriented as well as skill oriented syllabuses also we are incorporated based on the national education policy as well as industry needs based. We have created a wonderful syllabuses for the MSc marine biology courses. So any of the interested or any of the means neighboring, you just inform it. So we have the all the facility already. Previously they mentioned all the experts, as well as there are through the video that everything is a lot of facilities, a complete almost all the facilities we have in different aspects. I already mentioned the different aspects of facilities, even from basic to molecular tools, even next gen related equipments also we have. So any other interested or just inform these facilities are those things available. So please inform to others for joining that MSc marine biology course to learn research oriented, means technology oriented, as well as industry need based, as well as other advanced research outcomes also. So thank you, thanks a lot for your understanding. Anything clarification you just ask.
Thank you, sir. Thank you so much for your informative and elaborated presentation, sir. It was very insightful.
Thank you, ma. Thank you.
Yes. And there is some questions in the chat box, sir.
You just tell, ma.
Yes, sir. So one question is whether jellyfish is one of the bio indicators of organic pollution.
Yes, it's a bioindicator, no.
Yes, sir. And there is one more question. Whether venom protein mediated nanoparticles possess any toxicity.
As already mentioned, those based on the species venom only the toxicity are inducing, or that complex already mentioned the different species are different kind of complex of the venoms are there. So some of the species, so you can utilizing these drug delivery is highly toxic, but some of the species are very less toxic but activity also there, like when compared to cancer cell as well as the control cells also there.
Thank you, sir. Thank you so much. Now we are coming to the end of the session. And there are few announcements. We are delighted to share that admissions are now open for MSc Marine Biology program. This 2-year research-driven postgraduate program offers hands-on laboratory training, field exposure, and excellent research opportunities in marine science and biotechnology. We invite all interested students to register for the program or circulate this information to your friends. For updates on upcoming programs, workshops, conferences, and events, kindly visit the Satyabama website and follow our Center for Ocean Research social media platforms for the latest announcements. We also welcome researchers, faculty members, students, and industries to utilize the advanced characterization and research facilities which have analytical instruments that are available at Center for Ocean Research and National Facility for Coastal Marine Research. The brochures and the registration details are already shared in the chat box. You can take information from that. Thank you. Thank you, sir. Thank you for your wonderful information.
Thank you. Thank you. Thank you. Thanks a lot.